Hi everyone, welcome to NURS 1020 Anatomy and Physiology 2. In today's lesson, we'll be discussing Chapter 26, Fluid, Electrolyte, and Acid-Base Balance. Here are the topics that we'll be covering today. We'll start off by examining the various fluid compartments in the body, specifically ICF versus ECF, or intracellular fluid versus extracellular fluid, and then we'll move on through the three main topics, which is fluid or water homeostasis, which is intricately tied to sodium homeostasis. The electrolyte homeostasis that we'll be discussing are specifically related to potassium and calcium, and then we'll close off with acid-base balance by discussing the various buffers and the renal and respiratory systems that control the amount of acid in our body. Please remember to read Chapter 26 while always referring to the learning outcomes in the syllabus. Fluid balance is really water homeostasis. As we mentioned, it's tied intricately to sodium homeostasis, so that's what we're going to be discussing. Electrolyte homeostasis is the homeostasis of the major cations and anions. We're only going to focus upon potassium and calcium, the cations. Chloride is the main anion, but we're not going to focus on that. In terms of acid-base balance, it's really the homeostasis of the protons, which is the acid, and the bicarbonate ions, which is the base. We'll start off by looking at the water content in the body. In adult males, it's approximately 60% of your total body weight. In adult females, it's slightly less due to the typically higher fat content in females. Females usually, and this is just a generalization, have less skeletal muscle mass and consequently more adipose tissue. And adipose tissue has the least amount of water content. Consequently, females typically have a slightly lower water content relative to body weight. Also note that water content declines with age, usually due to the fact that there is less skeletal muscle mass. Here's the water content of the various organs within the body. It's not like you have to memorize that, but you can see, for example, the brain, 80 to 85% is water. Same thing with the lungs, the liver, blood, that would be plasma. That's about 50%. The skin also a very high content. On the other hand, bone has less water. It is mainly made up of calcium, phosphate, hydroxyapatite. So in terms of our total body weight, it's approximately 60% for a male and slightly less for a female, and you can see the various organs have a differing water distribution as well. Let's move on to the fluid compartments. We've seen this before in A&P 1, but just to remind you, total body water, or TBW, is equal to approximately 60% of body weight. I know that's for males, but we'll just assume that's the case. It's between 50 to 60. We'll just go with 60 for now. There are two main fluid compartments, ICF, or intracellular fluid. That makes up 2/3 of total body water, and ECF, or extracellular fluid, that's 1/3. In terms of the ECF, it can be subdivided into two compartments, plasma and interstitial fluid. So plasma is 1/4 of ECF, and if terms of total body water, it will be 1/4 times 1/3, or 1/12 of total body water. Interstitial fluid makes up 3/4 of ECF, or in terms of total body water, it's 3/4 times 1/3, or really 1/4 of total body water. Schematically, this is what it looks like. So within the cell is the ICF, and then the ECF is everything else, and ECF is divided into plasma, as well as the interstitial fluid, which is all the fluid in between the cells that is not located within the blood vessels. Here's it in a Venn type diagram. So extracellular fluid is approximately 2/3 of total body water, and ECF is approximately 1/3. Of the ECF, it's divided into two compartments, interstitial fluid, which is 3/4, and plasma, which is 1/4. Right, there's other fluids, such as in the CSF and so forth, but we're just going to ignore it. We're only going to focus upon plasma and interstitial fluid. In terms of the composition of body fluids, we all know that water is the universal solvent, and solutes are substances that are dissolved in water. All this is from high school chemistry. The solutes can be subdivided into electrolytes and non-electrolytes. Non-electrolytes are the organic compounds such as glucose, amino acids, even the lipids, urea, etc. They do not dissociate in water to form ions. Electrolytes, on the other hand, dissociate into anions and cations when dissolved in water. For example, most of the inorganic salts, all the acids and bases, dissociate when dissolved in water. It is also important to know that electrolytes have greater osmotic power than the non-electrolytes, and consequently have the greatest ability to cause fluid shifts between the compartments. Because in terms of water balance, it's really about the concentration, as we'll see, of sodium. Sodium homeostasis is directly tied to water balance. Electrolyte concentrations are usually expressed as millimoles per liter or milliequivalents per liter. The millimoles is the unit that is preferred in Canada. The milliequivalents is really related to the charge. For singly charged cations or anions, the millimoles per liter equals to milliequivalents per liter. The problem is when there's more than a positive charge or a negative charge, like for example calcium has a 2+ charge, then millimoles per liter is not equivalent to milliequivalents per liter. As I said, in Canada, the SI units are preferred and used. In other words, please stick with millimoles per liter. In the ECF, the major cation is the sodium cation, and the major anion is the chloride. In plasma, however, which is part of the ECF, there is a higher protein content and consequently a lower chloride concentration. So it's pretty similar in terms of plasma and ICF, except that in plasma there's a higher protein content. In the ICF or intracellular fluid, it's completely different. The major cation is the potassium ion, and the major anion is hydrogen phosphate. Just like in plasma, there is a higher protein content. In fact, the protein content is even higher than in plasma. Here's it in terms of a bar graph. So you can see the legend over here. ICF is cream colored, interstitial fluid is light blue, and plasma is red. So if you look at the concentration of sodium, we know this from A&P1, sodium is far more concentrated on the outside of the cell than the inside, very little in terms of the ICF, and most of it is located in the ECF in terms of plasma and interstitial fluid. Chloride, same thing. In terms of potassium, it's completely the opposite. All the potassium, or nearly all the potassium, is locked up inside the cell in the ICF, very little in the ECF. In terms of bicarbonate, you can see it's not as profound a difference, but nevertheless, most of the bicarbonate is located in the ECF. We're going to just ignore the rest of them, but just look at the protein content. Much higher within the cell. We know they are blood proteins, but in terms of the interstitial fluid, there's very little protein. Let's move on to discuss fluid movement between the compartments. This is also something that we've already discussed in A&P1 in terms of osmosis, but just to remind you that the osmotic pressure is what regulates the continuous exchange and mixing of fluids between all three compartments, between the ICF, plasma, and the interstitial fluid compartments. Water moves freely along the osmotic gradients in order to equilibrate osmolality. Osmolality is related to the protein content and not just the electrolyte. Osmolarity is just the electrolyte. So it's more accurate to talk about osmolality versus osmolarity. Nevertheless, for our purposes, we'll consider it synonyms of each other. A change in the solute concentration of any given compartment leads to net water movement. There's always water movement, but what we are concerned with is net movement of water. For example, when the ECF osmolality increases, water will ultimately exit the ICF and move into the ECF. The water exits the cell. On the other hand, if ECF osmolality decreases, water will going to move from the ECF to the ICF. In other words, the cell will swell. We've seen all this in A&P1, so please do revise it. This is just to refresh your memory. In terms of the actual numerical values, the osmolality is maintained at approximately 280 to 300 milliosmoles. That's the unit that's used. At equilibrium, ECF osmolality has to equal to plasma osmolality, has to equal to interstitial fluid osmolality, which has to equal to osmolality of the intracellular fluid. Everything goes to equilibrium. Specifically for water homeostasis, water intake has to equal to water output, which works out to approximately 2.5 liters per day. Our water intake consists of beverages, food, that's the water content of food because our food is made up of cells, and we know that nearly all the cell is just basically water. So there's definitely some water content in food itself, especially fruits and vegetables. Less so in meat because we know fat has a lower water content, as does muscle. And lastly, our water intake also is dependent upon the metabolic byproduct of aerobic respiration. Through aerobic respiration, we know when you take glucose and oxygen, you ultimately convert it to carbon dioxide and water. So let's not forget that water. We do get a certain amount of water from aerobic respiration. That's the metabolic byproduct of aerobic respiration. In terms of water output, we have insensible water losses through our skin and our lungs. That's not something that we can adjust, really, if we are not talking about perspiration. That is just evaporation from the skins and through regular breathing. We know the air is humidified, and so when we exhale, we're going to be losing some of that humidified air and consequently going to lose some of the water. when you exercise, you're going to perspire, and obviously you're going to lose more water that way in order to cool down the body, and we lose a certain amount of water in our feces. The rest of it is urine output, which is the variable part. The other parts are not very adjustable. They are more or less fixed. It's only the urine output that is variable depending upon our overall water homeostasis or balance. Let's discuss water homeostasis in more detail. When there's a rise in ECF osmolarity, that's going to be detected by the osmoreceptors in the hypothalamus. Why would ECF osmolarity rise? Well, because there's a decrease in the water content in the ECF. For example, severe diarrhea causes increased water losses, and if that is not balanced by water intake, there's going to be a net decrease in the water content in ECF, leading to a rise in ECF osmolarity. How is this manifested? It's going to be manifested by a decreased blood volume, decreased blood pressure, dry mucous membranes, and decreased skin turgor. In order to counteract the rise in ECF osmolarity, the third center in the hypothalamus is going to have to be stimulated. That in turn is going to increase the secretion of ADH. Ultimately, what we want to do is to increase the water content in the ECF, which is what we're going to be describing on the next several slides, and we've also covered this in some detail in the previous chapter when we did renal physiology. In the opposite manner, if there is a decrease in the ECF osmolarity due to a rise in the water content in the ECF, now we want to increase the osmolarity. How do we increase osmolarity of the ECF? By thirst inhibition and by inhibiting the secretion of ADH in order to decrease the water content in the ECF and ultimately increase ECF osmolarity. Here's it in a flowchart form. If there is insufficient or a net deficit of water in the body, that's going to be manifested by a decreased blood volume and an increase in the blood osmolarity. That's going to be detected by the osmoreceptors in the hypothalamus. Clinical manifestations we'll be describing in patho, for example, for clinical dehydration. The osmoreceptors in the hypothalamus, once they are stimulated by an increase in blood osmolarity, will stimulate the third center in the hypothalamus to increase our thirst sensation, which will cause us to drink more water, thereby decreasing blood osmolarity. We'll also talk about the effect of a decrease in the blood volume caused by a decrease in water content in the body. A decrease in ECF water content is ultimately going to cause a decrease in the blood volume. That's going to cause a decrease in the blood pressure, which will trigger the Renin-Angiotensin-Aldosterone System. Let's discuss ADH in more detail. ADH increases water reabsorption in the collecting ducts proportional to the ADH concentration. In other words, when there's a decrease in the ADH concentration, there's going to be a decrease in the water reabsorption in the collecting ducts, leading to an increase in water excretion and resulting in dilute urine. In other words, we are excreting out more water. There's going to be a net decrease in the volume of the body fluids, a net decrease in blood volume, for example. If there is an increase in the ADH concentration, there's going to be an increase in the water reabsorption in the collecting ducts, leading to decreased water excretion and more concentrated urine. The net result of an increase in ADH concentration means that there's going to be an increase in the volume of body fluids, specifically an increase in blood volume as well. How is all this detected? The hypothalamic osmoreceptors sense the ECF solute concentration and regulate ADH accordingly. Thus, if you want an increase in ADH concentration because of a decrease in blood volume, for example, a decrease in ECF volume status, then a decrease in the volume status is going to lead to an increase in the ECF osmolarity. That's going to trigger the hypothalamic osmoreceptors to secrete more ADH, which will lead to increased water reabsorption and more concentrated urine. Ultimately, the volume of the ECF will increase, resulting in a decrease in the osmolarity, and then ADH secretion can come back to normal. ADH release, please remember, is also stimulated by angiotensin II, released via activation of the Renin-Angiotensin-Aldosterone System when blood pressure decreases. Here's a couple of diagrams related to ADH. ADH is produced by the hypothalamus and then transported down the pituitary stalk to be stored in the posterior pituitary. ADH release is triggered by a rise in serum osmolarity and is also triggered by the angiotensin II, its release. What does ADH do? It inserts aquaporins into the collecting ducts, thereby increasing water reabsorption in the collecting ducts. Aldosterone is another hormone involved in water balance, water homeostasis. Aldosterone is released from the adrenal cortex and it's stimulated by angiotensin II. Aldosterone increases sodium reabsorption and hence water reabsorption in the DCT or distal convoluted tubules of the nephron. We've seen this in the previous chapter. Ultimately, what aldosterone does is it increases blood volume and thereby increases blood pressure as well. The two factors that affect aldosterone release from the adrenal cortex are serum potassium concentration. We'll discuss this a bit later on. Ultimately, aldosterone is one of the main hormones involved in potassium homeostasis. But aldosterone release is also triggered by the Renin-Angiotensin-Aldosterone System, and that occurs when, for example, blood sodium concentration decreases. It's really a decrease in blood pressure that triggers the Renin-Angiotensin-Aldosterone System. Ultimately, angiotensin II is produced, and then that triggers the release of aldosterone from the adrenal cortex. What does aldosterone do? Well, aldosterone increases sodium reabsorption in the kidneys, thereby increasing water reabsorption as well. Ultimately, blood pressure goes back to normal. We'll look at the effects of potassium and the role that aldosterone plays in potassium homeostasis a bit later on. Here's a review of the Renin-Angiotensin-Aldosterone System. Not going to spend too much here because we did all this when we discussed renal physiology in the prior chapter, chapter 25. A decrease in blood pressure will ultimately lead to the release of renin. Renin converts angiotensinogen to angiotensin 1. Angiotensin 1 is converted to angiotensin 2 through the effect of the enzyme angiotensin converting enzyme, mainly located in the pulmonary vasculature. Angiotensin 2 triggers the release of aldosterone. Aldosterone thereby increases sodium reabsorption in the DCT and hence water reabsorption as well. Ultimately, blood pressure increases and the renin-angiotensin-aldosterone system can be shut off, at least temporarily. A&P release from the right atrium is stimulated by an increase in stretch of the atria, and that's caused by an increase in blood volume. What A&P does is that it decreases sodium reabsorption and hence water reabsorption in the DCT of the nephron. Consequently, blood volume decreases and blood pressure decreases. You can think of A&P as the opposite of aldosterone. It does almost the exact opposite in terms of its effect on blood pressure. Lastly, in terms of sodium homeostasis, I just want to emphasize the difference between sodium concentration versus total sodium content. This is a bit tricky and we'll explain it more in class. The concentration of sodium is dependent upon the water content. It's really an indication of the amount of sodium relative to the amount of water in the ECF. That's what a concentration is, how much of sodium there is per unit of the solvent, which in this case is water. So it just depends upon the relative ratio of those two entities, the ratio of sodium relative to the amount of water. It's a relative ratio. It's a ratio. It's not the absolute amount that we're dealing with here. Consequently, the concentration of sodium really doesn't tell us about the total amount of sodium in the body or total body sodium. Why is this important? Total body sodium is ultimately, or the total sodium content is ultimately what determines ECF volume and therefore blood pressure. Because ECF volume is really blood volume and consequently blood volume affects blood pressure. So our blood pressure is ultimately determined by the total amount of sodium in our body. Consequently, one of the first line treatments for hypertension before moving on to the various antihypertensive drugs is to control sodium intake. That makes sense because what we are really after, what we are targeting is the total amount of sodium content in the body, because the total amount of sodium content in the body determines the ECF volume, determines blood volume, and ultimately blood pressure. However, this really has nothing to do with the sodium concentration. I know it sounds odd, but sodium concentration is a relative ratio of the amount of sodium relative to the amount of water. We'll discuss more of this in class. In terms of sodium homeostasis, there really aren't any sodium receptors in the body. So how do we know how much sodium there is? Well, that's why we measure blood volume and blood pressure. This is why there are receptors to measure blood volume. Really, blood volume is tied to the osmolarity of blood. They are osmoreceptors, and that is an indication of blood volume. As blood volume increases, blood osmolarity decreases and vice versa. They are also baroreceptors to detect our blood pressure. Those two mechanisms are what really tell us about the sodium-water balance. Since there are no sodium receptors, we have to rely on these secondary indicators. We're really measuring blood volume and blood pressure. And changes in blood volume and blood pressure trigger the various neurohormonal mechanisms that ultimately regulates sodium content. In other words, the sympathetic nervous system. When there is a drop in blood pressure, the sympathetic nervous system kicks into gear. That in turn will activate also the Renin-Angiotensin-Aldosterone System. Angiotensin 2 will promote the release of ADH. All that will increase water content, sodium reabsorption, etc. This is all linked to sodium balance, sodium homeostasis. And ultimately, sodium homeostasis is tied to water homeostasis. So please revise the effects of the sympathetic nervous system, the Renin-Angiotensin-Aldosterone System, ADH and A&P in terms of sodium homeostasis, in terms of water homeostasis. Let's move on to electrolyte balance. Specifically, we'll start off with potassium homeostasis. Potassium balance is controlled in the collecting ducts by regulating the amount of potassium that is secreted into the filtrate. A high potassium content in the ECF, in other words, a high serum potassium content, will increase secretion of potassium by the principal cells in the collecting ducts. A low potassium content in the ECF will decrease secretion of potassium by the principal cells. And yes, it is aldosterone that is the main hormone that stimulates potassium secretion by the principal cells. So how does this all work? Well, it's really based upon the fact that the adrenal cortex cells are directly sensitive to the potassium content of ECF, in other words, serum potassium concentration. An increase in serum potassium in the adrenal cortex in blood, which is present everywhere, including the adrenal cortex, causes the release of aldosterone. Aldosterone will then promote potassium secretion, right? So the serum potassium content, as we saw previously, is tied to aldosterone release. When potassium concentration increases, more aldosterone will be released, and then there'll be greater secretion of potassium by the principal cells in the nephron, and vice versa. If there's a decrease in serum potassium concentration, there'll be less aldosterone release, less secretion of potassium by the principal cells. Consequently, potassium concentration increases. In terms of calcium homeostasis, 99% of our calcium in our body is locked up inside our bones as calcium phosphate or hydroxyapatite. What role does serum calcium play in terms of blood clotting, as well as in terms of neuromuscular excitability? The role of serum calcium is very important. Parathyroid hormone, or PTH, is the main regulator of serum calcium concentration. When serum calcium concentration decreases, there is increased secretion of parathyroid hormone by the parathyroid glands. PTH, as we know, we saw in A&P1, increases osteoclastic activity. That's going to cause increased release of calcium from bone, thereby increasing serum calcium concentration. On the other hand, when serum calcium concentration increases, there's decreased secretion of PTH, leading to decreased osteoclastic activity. Ultimately, serum calcium concentration decreases. So PTH is the main regulator of serum calcium concentration. We're now going to move on and discuss acid-base balance. Acidity is really the concentration of protons, or H+, in our body fluids. Acidity is measured as pH, however, to avoid dealing with the nanomolar units of protons, because really you only need a tiny amount of protons and the units are just going to be so very small. So instead we convert the nanomoles of protons to pH, and pH is what we'll be using as a measure of acidity. Please remember from A&P1 that pH and acidity are inversely related. As acidity increases, pH decreases, and vice versa. The pH scale is typically from 0 to 14, although you can go below 0 and above 14. However, in terms of the bodily fluids that we'll be discussing, specifically blood, we'll see it's around 7.35 to 7.45. That's the pH range of blood. From A&P1, we also know that pH affects the functions of most proteins in terms of denaturation. When pH changes dramatically, many proteins become denatured. Consequently, they lose their three-dimensional shape and are no longer functional. If the proteins are no longer functional, for example, if the proteins were enzymes, then various biochemical processes are also going to be affected. Consequently, pH is very tightly regulated, especially in blood. For arterial blood, the normal pH range is 7.35 to 7.45. In venous blood, as well as in interstitial fluid, it's slightly higher due to the increased concentration of CO2. Inside the cell, ICF, the pH is actually neutral, pH 7, and that's because, as we'll see, due to the lower bicarbonate concentration. Bicarbonate plays a very important role in terms of overall balance. As bicarbonate concentration decreases, pH will also decrease. In other words, acidity will increase. Bicarbonate and the overall acidity are inversely related. As bicarbonate decreases, acidity increases because bicarbonate is a base. We'll talk more about alkalosis and acidosis in more detail in patho, but nevertheless, alkalosis is when the pH is greater than 7.45. And what's the difference between alkalosis and alkalemia? Alkalosis is a condition. Alkalemia is, as we know, a blood condition. So overall state is of alkalosis, but when we are discussing blood, we say alkalemia. Same thing with acidosis or acidemia. Acidosis is the overall condition. It has many manifestations, as we'll discuss in patho, but acidemia simply means that blood pH is lower than normal. In other words, arterial pH is less than 7.35. There are two types of acids produced in the body. Volatile acids, which is really just CO2. CO2 is the only volatile acid produced via aerobic respiration and is excreted by the respiratory system. The other class of acids produced in the body are the non-volatile or metabolic acids. They're called metabolic acids because they are produced as byproducts of metabolism. For example, the catabolism of protein that contains various phosphorus atoms ultimately releases phosphoric acids. Lactic acid, as we know, is produced from aerobic respiration, fatty acids, and other ketone bodies from fat metabolism. So those are all examples. Phosphoric acid, lactic acid, and ketone bodies are all examples of metabolic acids. For volatile acids, the only one is CO2. Before we move on to the actual regulation of acid-base balance in the body, we should discuss the buffering systems that are present in our body. Buffers are very important because they resist the change in pH, and as we saw, acidity affects the protein structure so much so that we can have various huge swings in pH on a second-to-second basis. Consequently, we need buffers. A chemical buffer is a substance that resists the change in the pH. There are three buffer systems in the body: the bicarbonate system, the phosphate buffer system, as well as proteins inside the cell. Bicarbonate buffers both the ECF and the ICF, however, it is the only important ECF buffer. What does a buffer do? A buffer resists the change in pH, and since bicarbonate is the only ECF buffer, it is very important to regulate the concentration of bicarbonate, and that is done by the kidneys. How is bicarbonate manufactured? Well, it is made from carbon dioxide or CO2, hence there is really an unlimited supply of bicarbonate that is available to us since we can easily produce CO2. The other two buffer systems are the phosphate buffer systems, which is very similar to the bicarbonate buffer system, and it is a very important buffer, the phosphate buffer system, in both urine as well as in the ICF. The protein buffer system, however, is the main ICF buffer, and that's because proteins contain both acidic and basic amino acid residues. When the pH drops, in other words, there are a lot of protons, there's a lot more acid, the basic amino acids in the proteins will bind the protons, thereby buffering the acid. On the other hand, when pH rises, there's a drop in the number of protons. The acidic amino acids will release the protons, thereby minimizing the change in the pH. For example, hemoglobin, which is a protein that we've discussed previously, we know the role of hemoglobin, but hemoglobin is a protein buffer of blood. In terms of pH homeostasis, ultimately the two organ systems in our body that are responsible for maintaining pH homeostasis are the respiratory and renal systems. They are the main regulators of pH balance. Please remember that a buffer cannot alter the pH balance. It can just simply resist the change in the pH. However, ultimately, if there's too much acid produced, pH will rise. What a buffer does is tries to minimize the rise temporarily, but in the long term, it cannot control the pH. What can control the pH are the two main regulators of pH balance, which are the respiratory and renal systems. They act far more slowly than buffer systems, but are far more powerful, as we just discussed, because they are the only systems that can really eliminate the excess acids or the bases and not just resist the change in the pH, which is what the buffers do. In terms of the respiratory system, it's all about the lungs. Our lungs eliminate CO2, which is an acid. Don't know how many times I'm going to have to say it, but it's essential that you simply memorize it. CO2 is an acid when dissolved in water. CO2 plus water will form carbonic acid, which will dissociate to form the proton, hence it's an acid, plus bicarbonate. If there's an excess buildup of CO2, there's going to be an excess buildup of acid. CO2 is a volatile acid, and that's why we need to excrete the CO2 the respiratory system is what excretes the volatile acid, which is just CO2. So it might seem really trivial, but all the CO2 that we produce has to be excreted. Otherwise, acidity is going to increase, pH of blood is going to decrease, because we know CO2 is dissolved in blood. There is a fixed amount, or rather a range of CO2 in blood, and that range, it's what will ultimately partly determine the overall pH of blood. If CO2 is not within that range, then pH is not going to be within that range, the pH of blood. So the respiratory system works toward eliminating CO2, which is the main respiratory acid, the main volatile acid. The kidneys, on the other hand, excrete the non-volatile acids that are produced by metabolisms that we saw previously, for example, phosphoric acid, lactic acid, ketone bodies, etc. The kidneys also regulate the concentration of bicarbonate. Ultimately, blood pH is determined by the ratio of CO2 and bicarbonate. It really is as simple as that. What is the ratio of CO2 and bicarbonate? Ultimately, through the Henderson-Hasselbalch equation, which we'll be discussing in class, that's what's going to determine blood pH. So just to remind you about the respiratory regulation of pH, when CO2 concentration increases, it means that we are producing too much of this volatile acid. pH is going to decrease. This is going to be sensed by the peripheral and central chemoreceptors, which will then stimulate the respiratory centers in the brainstem to increase our respiration rate. Our respiration rate will increase, thereby more CO2 will get excreted, CO2 concentration decreases, and pH is restored to the normal range. Why would CO2 concentration increase initially? Well, that's really due to hypoventilation. It's hypoventilation not in an absolute sense. It's hypoventilation relative to the amount of CO2 that is produced. Consequently, the solution is hyperventilation, as we just discussed. On the other hand, when CO2 concentration decreases, it means we have less of the volatile acid. pH is going to increase. This is going to be sensed by the peripheral and central chemoreceptors. The respiratory centers in the brainstem are going to decrease the respiratory rate. Consequently, less CO2 is going to be excreted. CO2 concentration increases and pH is restored to the normal range. Please remember that we need to keep CO2 within a normal range. It's not zero. The range of CO2 in arterial blood is 36 to 44 millimeters of mercury, right? So we cannot excrete out all the CO2. We have a certain amount that's dissolved in blood, but henceforth all the CO2 that is produced has to be excreted. But whatever is already dissolved in blood stays there. It's just that everything that we produce through aerobic respiration, after what's dissolved in blood, we have to excrete out. Otherwise, acidity will increase, and that will trigger the peripheral and central chemoreceptors to increase our respiratory rate to cause more CO2 to be excreted. So CO2 has to be kept in that normal range of 36 to 44 millimeters of mercury. When pH decreases, that's acidosis due to a buildup in CO2 concentration. That's going to stimulate the peripheral and central chemoreceptors to increase their respiratory rate by stimulating the respiratory centers in the brainstem. More CO2 is going to be excreted out. Blood CO2 concentration is going to decrease. pH is going to increase. We'll look at the role of bicarbonate later on. On the other hand, if there's less CO2 dissolved in blood, that will be alkalosis or respiratory alkalosis. The peripheral and central chemoreceptors will be stimulated to decrease the respiratory rate through the respiratory center in the brainstem. Less CO2 will be excreted. Blood CO2 concentration will increase. That's going to ultimately cause a decrease in the pH as more CO2 is excreted out. Ultimately, acid-base homeostasis is restored. Again, we'll look at blood bicarbonate concentration a bit later on. Let's now discuss renal regulation of pH, or the role of kidneys in terms of acid-base balance. The various metabolic or non-volatile acids that are produced through regular metabolism in the body can only be excreted by the kidneys. Remember that the lungs can only excrete out the volatile acid, CO2. The non-volatile metabolic acids can only be excreted by the kidneys. This is accomplished by regulating bicarbonate concentration. Therefore, the kidneys have to reabsorb all the filtered bicarbonate, which we haven't discussed before, as well as generating new bicarbonate. It is important to note that the reabsorption of filtered bicarbonate does not affect proton secretion, acid secretion. To secrete acid, the metabolic acids, new bicarbonate must be reabsorbed. This might all sound confusing, but when we look at it in diagrammatic form, it'll become hopefully a lot clearer. The secretion and ultimately excretion of protons have to occur via two mechanisms, either through a phosphate or ammonium buffer system in urine. Here's a diagram from the textbook that explains the reabsorption of bicarbonate. It turns out that there are no receptors that can ultimately reabsorb bicarbonate from the filtrate. Remember, at the glomerulus, everything is filtered. We already talked about the reabsorption of sodium and then water follows sodium, etc. But what about bicarbonate, which is also filtered? This is how bicarbonate is reabsorbed through an indirect mechanism. We use the equation that we previously seen, whereby CO2 plus water gives you carbonic acid, and then it dissociates to bicarbonate and proton, except we do it in reverse. So now we have the bicarbonate that we want to ultimately reabsorb into the bloodstream. How do we do that? Well, we convert the bicarbonate to CO2. For that, we need a proton. We'll see where this proton comes from. That will then automatically form carbonic acid, which in the presence of carbonic anhydrase enzyme, which is on the brush border of the tubules, will form water and CO2. CO2 will then diffuse into the cells of the tubules, whereby the reverse reaction will occur. CO2 plus water in the presence of carbonic anhydrase will form carbonic acid, which will dissociate to give you bicarbonate. The bicarbonate is reabsorbed. This is the roundabout way of getting bicarbonate into the cell, and then ultimately it's reabsorbed into the peritubular capillaries. The proton that's generated will be secreted out through the sodium proton exchanger to ultimately react with the bicarbonate in the lumen, right? So this proton doesn't just magically appear, it's a cycle. We're not really producing it, well we are, but then it is consumed. So there's no really net change in the pH. That's why we said that the reabsorption of bicarbonate doesn't really alter, it's not really not involved with proton secretion, because this proton that is secreted was generated from the CO2, which we had converted the bicarbonate to, right? So you generate the proton, then the proton's used up. There's no net production or consumption of the proton. This is the reabsorption of bicarbonate, which is very important because we need to regulate the concentration of bicarbonate in blood. Otherwise, you're going to alter pH balance. So this is just a reabsorption process. However, unfortunately, there isn't a diagram in our textbook that explains this. If you need to secrete a proton, now you need to generate a new bicarbonate, which will then be reabsorbed. So here's the secretion of the proton. Where does this proton come from? Well, the metabolic acids, either lactic acid or phosphoric acid, ketone body, it doesn't really matter. It's a metabolic acid that we secreted actively. That's why you have this ATPase here. It's a primary active transport that secretes out the proton. The thing is that we need to buffer it, otherwise urine will get very acidic, and that's going to mess up a lot of things, which I'm not going to go into detail. It'll mess up all the reabsorptions of all the other nutrients and electrolytes, so we need to buffer it. We buffer it either with this hydrogen phosphate, or alternatively with the ammonium ions over there. But in either case, we need to buffer it. But the point is that this proton, where do we get this proton from that we've secreted out? Well, it comes from CO2, ultimately, and that CO2 in the presence of water and carbonic anhydrase will form carbonic acid, which will then dissociate to form the proton and bicarbonate. The proton is secreted. Ultimately, we're secreting a acid, the proton, thereby we are increasing pH, we are lowering acidity. This bicarbonate is reabsorbed. This is what we talk about. So the secretion of a proton is tied to the reabsorption of a new bicarbonate. This bicarbonate was not filtered as we saw previously. It's not in the filtrate. It's new. This is new new bicarbonate that is generated and reabsorbed over there. So that's how the kidneys ultimately excrete out the excess metabolic acids in our body. It's tied to the reabsorption of new bicarbonate. That brings us to the end of today's lesson. We started off by looking at fluid or water balance, which we know is directly tied to sodium homeostasis. We moved on to electrolyte homeostasis, specifically focusing on potassium and calcium. And then we finally closed off by looking at acid-base balance. There are two types of acids, volatile acids, that are regulated by the lungs. The lungs will excrete out any excess CO2. And then we have the metabolic acids that are regulated by the kidneys in terms of regulating bicarbonate concentration. In our next lesson, we're going to be covering Chapter 27, the reproductive system. Thanks everyone. Have a wonderful week. Bye.